Hello and welcome to the Tillage Edge with me, Michael Hennessy. This is your regular update for all your tillage news and advice. It's heartening to see scientists have rapidly developed numerous vaccines against COVID. This high level of science is employed on a day-to-day basis in the agricultural world. And as with vaccines, there are many who readily accept these outcomes and others not so easily. Plant breeding has advanced hugely in the past 50 years, from targeted breeding to genetically modified crops and now gene editing, all of which are having an impact on agriculture worldwide. We are joined today by Ewen Mullins from Oak Park to get an under-the-hood look at plant breeding and biotech developments. I first asked Ewen about the types of plant breeding which have been developed over the past 200 years or so.
Okay, Michael. So it was back in the 1800s when an Augustinian monk in Austria by the name of Gregor Mendel discovered basically that when you crossed two varieties, the siblings or the progeny had traits or characteristics similar to the parents. And most importantly, those traits were inherited in a particular ratio. And that really, he was working actually on pea. And by crossing, for example, a red flower with white flower, he discovered that some of the progeny were red, some were white, and some were a mix of both. So that really was the start of it in terms of inheritance. And you come forward 100, 150 years, what we saw in the early 1900s was a rapid acceleration in people researching and looking at the ways to make plants yield more, yield better. And one of the techniques that was adopted there was what's called mutagenesis, and was what, out of interest, that actually came out of the Manhattan Project, or was a spin-off from that, because what they discovered was when they were blowing up all these nuclear bombs for testing and stuff out in the Arizona desert and Nevada, they realized that a lot of the plants that could be maybe 20 miles or 30 miles away from ground zero were showing very different traits. And they were looking stunted, they were looking mutated. And what they realized, and very quickly, was that you can use mutations to change how certain traits appear in plants. So what you're doing is you're changing the DNA. So, I mean, that's really been the bedrock for the last 40, 50 years in terms of breeding new varieties. Using mutagenesis, be it chemical or physical, for example, X-rays or other forms of radiation, or you can go back to the traditional way, which has been there for thousands, 10,000 years, which is transferring pollen from one flower to another, taking the progeny and growing them up and seeing do they have the particular traits that you want, be it disease resistance or shorter stem or more grains per ear, etc. But that's really a slow process. It takes a year, obviously, before you get a result.
So just with that, then, Ewen, natural selection is obviously relatively slow, as you say, and GM then, as in genetically modified, is obviously a different process, like you were talking about a second ago. How much quicker or much better is GM in comparison to natural selection?
So basically, as you say, natural selection just takes time, and it's nature basically selecting out the bad fruit, the bad seeds, the bad progeny, so that the survival of the fittest pertains. And that's what evolution is. So you go back to Charles Darwin. He was the first person to put his finger on it and realize that actually only the fittest of the species, be they plants, animals, whatever, were surviving the pressures and the stresses that they were encountering in their local habitat. So natural selection now, of course, nowadays we use DNA fingerprinting techniques. So what does that mean? Basically, every gene has its own DNA fingerprint. And using technologies that have been, invented and developed in the last 20, 25 years, it's possible to have a DNA fingerprint for every particular variety of a plant. So that can speed up natural selection a bit. So for example, if you wanted to make a new wheat variety or a new barley variety, it takes approximately 10 years from the moment you make the first cross. And the cross is taking pollen from one plant and transferring it onto the flowers of another. So the plants that will be grown in 2030 are the ones that will be derived from crosses made this year and next year. So it's very important you get your parental material correct. So, but as I said, it just takes a long time. You're looking at, approximately 10 years for cereals, about 15 years or 13 to 15 years for potatoes. When it comes to GM techniques though, what you're doing is you're accelerating the process greatly. So while it might take 12 to 13 years to produce a new potato variety by crossing two parents, say, for example, Rooster with Maris Piper, if you were to use a GM technique to improve the performance of a particular variety, say at Maris Piper, you wanted to make it blight resistant or more resistant to drought, for example, you could change the genetic makeup of that plant using a GM technique, and that would deliver a plant for you, a new variety, in probably about 6 to 9 months. Now, that variety then has to go through all the standard regulatory testing and evaluations that a new variety would have to do. So you'd add on another, 3 to 5 years on top of that. But basically, GM as a technique is a way to accelerate the breeding process, ideally probably cutting it in half.
Okay, so before we get into maybe a little bit more in depth into the GM side of the house first, but just to concentrate a little bit on the types of crops that GM is suitable for. I've never heard necessarily of any barley or wheat or oats being a GM, or the possibility of Irish farmers potentially growing them as a GM. It's all maize or cotton or soy or something like that.
Yeah, no, you're right. And I mean, and it's really been focused, I suppose, since the first GM plant was actually a tomato back in '96, with the Flavor Savor tomato, which had a longer shelf life. And when it was first generated, obviously it was the producers and the inventors at the time felt that it would revolutionize food waste because the material would last longer, so the consumer has longer to consume it, basically. However, there was a backlash to it, both in parts of the US, in North America, and in Europe. And a lot of that was driven by, obviously, fear of a new technology and the unknown, which is not extraordinary and is understandable. But what happened then was the main companies that at the time, the ag biotech companies, decided to shift their efforts and focus on the large commodity crops, effectively the cash crops, the soya, the maize, and as you said, cotton, and to a lesser degree, rice. And there was a lot of patenting frameworks and intellectual property systems put in place to kind of ring fence the application of the technology to those main commodity crops. And that drove the development of GM acreage through the late 90s into the 2000s. And today we primarily still have soya, maize, cotton. You also have canola or oilseed rape. And then I suppose in recent years, we've seen things like GM papaya in Hawaii. We've seen GM eggplant in parts of India. And there's also other crops such as GM rice and GM banana in trials in parts of Africa and Asia as well. So it is expanding primarily because a lot of the patents are now coming off, are coming off patent. So the regulatory, or sorry, the legal restrictions on the use of the technology have loosened up.
So it's limited by patents to a degree rather than crops.
It was initially at the start, and also one of the ways to modify a particular plant is it's all based primarily on using a bacteria called agrobacterium. And this is a naturally occurring bacteria. If you're walking through a forest and you'd see them in many trees in Ireland, you'll see a kind of a tumor or a mass of cells, a growth sticking out the side of a tree. That's driven by agrobacterium, which is a soil pathogen which infects plants, a lot of plants, and it transfers its own DNA into the plant. So the plant then starts to produce proteins and goodies, sugars that feed the bacteria. So in effect, it's a bit like a parasite, but a parasite would typically kill the host. Agrobacterium has no interest in killing its host, it just wants to live off the host. So it was back in the 90s when scientists discovered, well, if we were to substitute the DNA that it transfers into a plant with, for example, a disease resistance gene or a herbicide tolerant gene, well then could we make the plant cells resistant to the herbicide, for example? So that's what happened. Now the thing is that agrobacterium as a pathogen in itself, it elicits a defense response from a lot of plants that have evolved to resist it. And that's why, as well, in addition to the legal and the IP side of things, is that certain plant species are more amenable to the use of agrobacterium than others. But in the last 20 years, an awful lot of work has gone into addressing that sort of work. Okay.
So if you go into the GM techniques, are there different types of GM techniques, or is it all pretty much the same thing?
No, there are. And I suppose like any technology, you compare it to a smartphone. If you asked a teenager nowadays, would they be interested in an iPhone 3 or an iPhone 2? they'd look at you weirdly because they wonder why in God's name you'd want to even contemplate using something like that. And similarly with a lot of biotechnology applications, they're constantly evolving. So there's actually like two parallel streams going on. There's the application of the techniques to various different crops, and parallel to that, there's people working on making the techniques more efficient and better and easier to use. So since the first techniques in the mid-90s, that really started out with what's called transgenics. And that's taking a gene from a species that would never naturally mate or cross with the target plant that you wanted to change. So for example, if you wanted, golden rice is a great example where they took genes from daffodil and put them into rice to increase the vitamin A content of rice, to alleviate vitamin A deficiency in populations of Southeast Asia. But like a daffodil and a rice plant are never going to cross. So that's going to need GM techniques. More recently then, there's a development of what's called cisgenics. And cisgenics is transferring a gene from one species to another species, but those species are related. So in effect, it's wild potato to conventional potato or wild apple to conventional apple. And that can happen through natural pollen transfer. But I mean, if you transferred pollen from a wild potato to a conventional potato to get a new variety, you're looking at probably 25, 30 years plus before you're going to get a variety that would meet consumer needs and demands. And then more recently, of course, there's gene editing, which really has been seen a monumental change in the application of breeding techniques.
Okay, so if we look at I suppose the majority of what's imported into Ireland, let's just say for feed for soya and for maize, what sort of GM technique is being used on those crops?
They would be primarily transgenics, because the majority of that material, actually the overwhelming majority of that material, the maize, is herbicide tolerant. So they've taken a gene from bacteria and they've put it into the soya or the maize to make it resistant to maybe dicamba or glufosinate or glyphosate herbicides. In addition, what they're doing now is they're actually stacking multiple traits in the same variety. So you could have a variety that is resistant to 2 or 3 herbicides because it has 2 or 3 individual genes to make it resistant to the corresponding herbicides. And then they would also have put in there maybe 2 or 3 insect resistant genes, so Bt genes, which make them resistant to particular lepidopteran insects, that caterpillars and stuff like that, can become quite serious pests on maize in the US and Canada.
Okay, and obviously those crops, they're a very big part of the imports into Ireland. Do you have any idea what sort of percentage that is in terms of GM versus non-GM?
I don't have up-to-date figures. At the moment, for every 5 tons of animal feed we use in the country, we produce 1 ton domestically. Now, back in 2008, we were importing about, I think it was about 1.5 to 2 million tons of feed, and approximately 50% of that was GM. Now, the importation of the GM feed obviously is driven by the fact that the majority of production of these commodities in North and South America is with the use of GM varieties. So that's why they're being imported. And they go through a rigorous assessment process through EFSA, European Food Safety Authority, to make sure that they're safe, both for animal feed use and they end up obviously in the human food chain.
And Ewen, just in terms of you mentioned transgenics and cisgenics, and perhaps the cisgenics because it's are related plants that are using to transfer the genes and potentially, I suppose, maybe more acceptable. Does legislation or legislators treat them any differently from the point of view of one is GM or one's not so much GM, or are they both the same?
No, from a legal perspective, they're both considered to be GM. But what's interesting is the Eurobarometer survey, which is basically consumer surveys done by the European Commission every year across various different aspects of society, looked at a survey, I can't remember, I think it was 2011 or 2012, where they surveyed consumers and they asked them a question about cisgenics versus transgenic in terms of acceptance. But I think there was a 61% acceptance of cisgenics as opposed to about 30% acceptance of transgenics. And that was quite insightful at the time and very revealing, because it shows that people are more willing to accept the acceleration of a natural process rather than the development of a new process, which is what transgenics would be in itself.
Okay, well, look, that's, I suppose, progress to a degree. I mean, everything may be in stages, and people don't take change, I suppose, lightly, and they stick with what they know. But coming back just a little bit, I know here in Oak Park, we had some trials. I think you were running trials in terms of GM on potatoes. You might maybe just give us a quick overview on what was involved there and how that worked out.
Okay, so that was basically, we were evaluating a cisgenic potato, which had been developed by colleagues in Wageningen University in the Netherlands. And this was part of an EU-funded project called AMIGA, when the title was Assessing and Monitoring the Impacts of GM Crops on the Agro-Ecosystem. So it's a bit of a mouthful, but basically what we had synchronous trials here in Oak Park in Carlow, as well as trials in Wageningen in the Netherlands. Because in the Netherlands, whereas we think we have problems with late blight, their problems are far more severe. So they, just like us, are heavily reliant on the use of fungicides. So the cisgenic potato variety that we used and we evaluated had been generated through the use of a cisgenic technique. So it took a gene from a wild potato, and that gene conferred immunity to late blight disease, Irish strains of late blight, and also immunity to Dutch strains of late blight. And we know this because over the 3 field seasons that we evaluated it here in Oak Park in the field trials, we saw that it reduced the need for fungicides from a typical annual usage of 13 sprays down to 1 to 2 sprays per season. In addition to that, we looked at environmental bioindicators, which are organisms that tend to be very sensitive to disturbance to the soil or any change in management in a crop. So that included fungi, bacteria, little plant nematodes, and also colleagues in the Netherlands came over and they looked at above ground fauna diversity as well. So what we saw was that the environmental impact of just growing potatoes was reduced by over 90%. And this is very significant because we're looking at farm to fork goals out to 2030, where we're being told to reduce inputs by 50%, the use of PPPs, plant protection products. So this particular technique has the real potential to reduce it far more than 50%. So it is a very significant technique.
So Ewen, the impact, as you said, there on non-target species, if you like, or flora and fauna in the field, was that in a very short term, or was that a little bit more longer term?
So that was done over the 3 years. So they were looking at, they would have different traps at ground level and approximately 2 feet above ground. And they were taking 3 to 4 sampling times through the growing season. So we were trying to capture as many bioindicators, and as you said, NTOs or non-target organisms, as we could, to see was there any difference between the 3 treatments in the experiment, which was basically the cisgenic line, which had no fungicide, it just had that single wild potato gene. We then had the original variety, which was Desiree, which had fungicide, and then we had the original variety Desiree, which had no fungicide.
Okay. So and then on the other side, I think at the house in terms of research, I know you've been working very intensively in this GM area, I suppose in the lab, maybe more so out in the field. And you came up with an insiver, I think is what you call it, in terms of an alternative way of GM. You might just maybe give us a very brief outline of what's involved there and how exciting, I suppose, it might be for the future.
So earlier on, I mentioned the fact that Agrobacterium elicits a defense response from certain plant varieties and plant species because it is a natural pathogen. So that's one of the reasons why not as many species have been transformed or engineered using GM techniques. So we were curious to find out, would there be any other soil bacteria out there that maybe could transform and engineer plant species, but wouldn't have the restrictions that Agrobacterium had. So it was very basic blue skies research, and through many long hours of PhD students and the postdocs that worked on all of that work, we were able to identify one particular strain called Ensifer adherens. And Ensifer lives naturally on the roots of plants. We took it off the root of an oilseed rape plant, and it has shown the ability to transform more varieties within individual species than Agrobacterium does, especially actually in crops like potato, We've also looked at oilseed rape, safflower, barley, and a few other different types of species, rice as well. And we've had a lot of interest in it around the world because basically, obviously, it overcomes some of the limitations of agrobacterium. It's not as efficient as agrobacterium. So while it will engineer a broader range of varieties, it doesn't necessarily engineer to the higher rate that agrobacterium will do. So that's why it just needs more research and development.
Okay, geez, a very exciting and cutting edge type research there. We mentioned CRISPR technology, or you mentioned CRISPR technology a little bit earlier, or gene editing, if you like. We're not necessarily going to go into it now because we don't have enough time today. But do you think that the CRISPR technology will be a part of crops in the future in Ireland?
Not in the near term, because in 2016, I think it was, the European Court of Justice basically made a ruling that gene editing was a form of GM application and GM technology. So as a result, anything that is edited and is to be grown in a field, be it for commercial or field trialing, has to go through the current legislative testing and regulations that are required, and that's quite a time-consuming and expensive process. So that has effectively put a brake on the development of novel crops across the whole of Europe using gene editing techniques. And this is in contrast to other jurisdictions outside of Europe, which are forging ahead with the use of precision breeding to develop new varieties. Well, actually, it's not even new varieties, I suppose. It's to enhance existing varieties. So they might have a particular rice plant, a rice variety in China that performs really well, but is susceptible to mildew. So they've edited it, and now that particular plant is basically, that variety is the exact same, just with a single change to one gene, and now it's mildew resistant. So the potential benefit of editing is huge, but within Europe, we do need to have serious discussions about its application.
Okay, Ewen, thank you very much for your very insightful knowledge, and obviously there's a huge amount in this area, and hopefully farmers will see the benefit of it shortly. I'd love to invite you back next year, and we will go through maybe the CRISPR side of the house in a little bit more detail, because that's probably more the future than GM potentially here in Ireland. So with that, Ewen, thanks very much.
Thanks, Michael.
That's it for the Tillage Edge this week. And my thanks to Ewen for joining me on the podcast. Don't forget to rate, review, and subscribe on Apple Podcasts or Spotify so you never miss an episode. And for more farming information, go to teagasc.ie. I'm Michael Hennessy. Thanks for listening. I'll be back in 2 weeks with more tillage news and advice.